Welcome to the virtual HOAHub workshop on how to analyze your HiP-CT images of human organs. My name is Dahl and I'm a professor in image analysis from DTU Compute, and I'll give the first presentation. So the segment anything model is a so-called foundational model for photos, and it does a remarkable job. It can, when you upload an image to the web app, you get very accurate segmentation, and you're just thinking, wow, can this really be true? But this is also a model that's trained on 11 million images and more than 1 billion labels. So it requires also a tremendous amount of data to get to this level. And it's not segmenting structures that we would typically see from the human organ atlas data. Another area is the medical image analysis area. And they're actually working with 3D data. So progress has also been made there based on the advances for segment anything model, namely for example, the MedSam segment anything in medical images. That's a model trained on 1.5 million images from 10 different modalities. When you look under the hood, you realize that what they've done for the volumetric data here is that they take out slices and then they make them 3-channel because it needs to fit into the architecture and the framework of the segment anything model. But nevertheless, quite impressive results are obtained by the MedSam model as a user interaction segmentation model. And thinking about human organs at the level of a clinical CT scanner is of course limited in resolution. This is also reflected in the work done in, for example, this total segmentator model, which is another quite impressive model where you segment out structures of the entire human body. So there's for training this model, it's based on a unit, and it's trained on 1,200 approximately CT scans of the human body. But within each of these scans, there's 104 anatomical structures labeled. So it's also a huge effort that has went into this. And this data, based on this model, if you should do this segment a person manually, it would take forever. But with this model, you can there's an online demo showing how it works, and I've just recorded a video of this. It's a speed up video. It doesn't go this fast. Perhaps it takes 5 minutes, but after 5 minutes, you can download the result and then load it into a visualization tool, and then you can see all the structures segmented out. If you had to do this by hand, it would take forever. It's like several days of work to get this result. So it's quite impressive. But on the other hand, this type of data does not have the same kind of variability as you would see in micro CT data of human organs. So therefore, what can we learn, and how can we use these progress that has happened in the field of AI and segmentation? I think that with this human organ atlas data, so we are in a situation where we record data at a scale that has not been done before. We get entire organs at extreme resolution. It's a special kind of data with the hierarchical structure where we have the overview scans and we have zoom-in scans. It gives really large images and great details of great complexity. And also a kind of data that we are not so used to working with. And then we have the requirement that data should be shared. We also, it's not as constrained as CT scans of the human body, but nevertheless, it's not like any type of data. It is actually HiP-CT data that's collected. So it's the same type of structures, you could say. So there's some constraint to what is possible to see and what's not. So what about we work towards sharing not only the data, but also the models that have been used to segment the structures and analyze the structures, and also especially sharing annotations, because deep learning-based segmentation requires annotations. And for this type of data, you typically also need to have experts involved in the annotation phase. in order to get the structures right. But if we collect this in a way and do it structured, we might have the basis for training larger models and start working with foundational models for this type of data. And I think this is a very exciting perspective. And this is the theme that I hope we can discuss at the physical meeting in this working group that we will have in the fall, because there's both something about the standards for how to collect the annotated data, how do we share it, how do we make it into a format such that it's easy to train new models based on this, and how do we actually make these new models useful when we have segmented one structure in one type of images and then want to segment other structures in other types of images. Is it even possible? And this is exciting research from an image analysis point of view. So with this, I'll go back to this workshop because the purpose is not to discuss this. It's of course fine to have some discussion in the end of this, but it'll be the theme of the next workshop. For this workshop, we will make a status of what's going on the image analysis side of the Human Organ Atlas hub. And for this, we have invited a number of speakers. They are primarily the ones that are doing the work. And therefore, I'm looking very much forward to hearing all the presentations of today. And we will start with Hugo from his work on the analysis of the colon, which is done at the University of Copenhagen.
Nice. Okay, I'm just setting up my windows. Okay, that's fine. Do you hear me well? Oh, I don't hear you anymore. Okay, this is okay because of the microphone. Okay, so good morning, everybody. Thank you for giving me the opportunity to present my work on CT scans. So yes, I'm not actually a PhD student. I'm a master's student from EPFL, and I'm doing my master's thesis here at the University of Copenhagen under the supervision of Jannis Schaeper. And I'm working on the 3D reconstruction and vascular analysis of colon CT scans. So just to give you a short context of what I'm doing, why I'm doing here. I did a bachelor in mechanical engineering at EPFL, so in Switzerland. That I moved in robotics and data science, was at EPFL. And amongst other projects, my taste for the medical field came from my internship at Medtronic, where I did computer vision. And since 2 months now, I'm doing my master thesis here. So working on colon CT scans. So why this analysis? It's important to know that the colorectal cancer is the third most common cancer worldwide, and it's accounting for approximately 10% of all cancer cases, and it is the second leading cause of cancer-related death worldwide. So here in Denmark, we have like 2,000 surgeries of this type each year, and amongst those 2,000 surgeries, there are 5% of rejection, and each rejection necessitates rapid intervention to save the patient. So the underlying hypothesis of that is that the surgeons may have an incomplete visualization of the vessels, and then they are likely to cut some of them during the surgery. Then as a result, the vessel density is decreasing and may poorly irrigate the colon, then causing the rejection. So what kind of data we have? So the data are the CT scans at different scales of a colon from a cadaver in France. The tissues around the colon were removed. We don't know exactly how. Maybe it's we don't know if it's like properly cut or roughly cut. And the different scales are shown on the right. They are ranging from 1 micrometers to 40 micrometers. And the lower scales are basically a zoom on the higher scales. And we can see that it's not well contrasted. So the challenge of this data set is mainly to handle the amount of data we have and the memory, for example, for 3D rendering. So we basically have stacks of images ranging from 2200 to 9600 of images, and the resolutions are ranging from 1900 squared to 5500 squared. The other challenge is to segment the different parts of the column for our future statistical analysis of the vessel density. So our first intuition was to threshold the images in order to extract the column structures and the column from the background. So for that, we used the K-mean algorithm. It was a good candidate, but it provided noisy results, and so we were not able to move forward with that. And we also created masks by just applying image enhancement and morphologies to better extract the region of interest. But yeah, we didn't move forward with the K-means, but we kept the masks to help us create our 3D visualization. So the first 3D visualization is the higher scale. So this is the 40 micrometer scale. This is actually the full column, but because of the memory constraint, we have to take every 4 images and downsample each image by a factor of 4. And after that, we were able to apply a multiple HOAHub threshold with 6 different thresholds. So to have a better understanding of what's inside without downsampling images, we can see here a local rendering of the 40 micrometer scales, and we can see better the small vessels. So yeah, this time is not downsampled, and we also applied HOAHub threshold. For the lower scales, it was a bit trickier because data are much more noisy and are very low contrasted compared to the higher scales. So we can see here on the first image, there are also huge difference of intensity between close slices. So we need a bit more pre-processing to produce a more suitable rendering, and it's not even clean for now. But this is the rendering with 2000 images, and all images are downsampled 2 times. So on a video, it's giving that. So here it's now downsampled. It's a sample of 100 consecutive images, and always with our HOAHub threshold. And all the 3D rendering were made on TomVis software. Okay. Now to move forward with the vessel density analysis, we need to segment the different parts of the column. So a first ground truth with 10 clusters was provided by a doctor, but it is the only one, and it's not even the full image. So it was a bit tricky to move forward with advanced segmentation techniques like deep learning and neural networks. So as expected, the very small amount of data gave us poor performance by training a unit model, even after splitting into patches, doing some image augmentation or other tricks. And we also tried pre-trained models like the segment anything model, but it was not better than that because of the complexity of our images. Yeah, you can see the images here and the predictions. So we can see that the region on the raw image are very hard to get. Yeah, it was not that good. And for the next step now, we are just looking to create more ground truth using a semi-supervised segmentation with the MED-SAM model. So the goal is now to simply segment the region of interest from the background, so to have 2 clusters such that we can move forward with the model development and learn how to handle the images and their specificities. And later, it will be easier to refine the model when we will have more refined segmentation for more images. And yeah, we really need these steps because it will allow us to isolate and analyze the vessel density. And yeah, that's already all for me. So thank you for your attention, and if you have any question, I'll be happy to answer them.
Thank you, Hugo. I think it was a very nice presentation. So let's open for questions. Let's see.
If there is no question, that's fine.
Yeah, there will be questions. Otherwise, I will ask the first question.
Okay.
I'm just giving a little bit of time. You know, there's the 30-second rule saying you need to wait 30 seconds for anyone to react. But let me start with, oh yeah, we have Tom O'Mahony.
Omani, can you hear me okay?
Yes, we can hear you.
Okay. I'm just wondering, you said you tried the k-mean segmentation. Have you tried, which is a very hard clustering method, have you tried more fuzzy clustering, such as local c-means or anything? Because that might
like the fuzzy c-means algorithm.
Yeah, local c-means. So k-means is a hard clustering one. But I know some other groups have had some success, not necessarily with colon things, but with c-means, local c-means, which is related.
I tried this one as well. It was a bit difficult to implement in my on my side, so I haven't moved forward that much with that. The first result I got were not that good, so that's why I didn't dig deeper in this. In this sense.
Fair enough.
In this way, but yeah, maybe I should go deeper in that way.
Yeah, it does. It does. When I've used it, it does require a bit more fiddling and sort of tuning than basic k-means. But if you found it was not really giving you anything more than k-means in your initial ones, it probably won't, even with lots of fiddling.
That's a good point. Thank you.
Thanks.
I will learn that.
Yes, David.
Well, hi. So if I quickly introduce myself, I'm the Human Organ Atlas data scientist based in Claire and Peter's team. So it's kind of my job to make sure the data is usable, accessible for people. So I thought I'd just say hi to everyone. Really nice talk. I'd just say we'd love to at some point as the Human Organ Atlas go through and have like these whole organ segmentations for all the data we release. So I was just wondering if you had a sense of like how good your the thresholding segmentation was, and also how fast it runs. Presumably it doesn't take too long.
Oh yeah, you mean the O2 threshold I just
yeah
for the full column, so it's about like 2000 images. I think it's about like 7 or 8 minutes to run, and it's pretty accurate, but it depends mainly on the quality of your images and the contrast you have in. But you can always do pre-processing to manage that, but it's a bit difficult to handle the different scales we have because as you saw before, the lower scales are harder to segment than the higher scales. So it really depends on the quality of the images and the contrasts you have on it.
Oh, brilliant! Thank you.
Yes, Claire.
Hi, Hugo. Thank you for the really nice talk. Just one question. I was wondering, I know it's really, really hard to even get off the ground with the UNet segmentation models when you have such a small amount of training data. And I wondered if you'd had a look at any pre-trained models potentially. I don't know what might be out there that's trained on like CT of colon, like clinical CT of colon, and if that might provide any use as just like a pre-trained model to see if it performs any better than something trained from scratch.
Yes, it's a good point actually. I tried to start with that, but the pre-trained model are very general and are not very specific to that task. So I haven't found a model for specifically CT scans, and especially at those scales. So yeah, as you may have seen, the region are very tricky to get. So pre-trained model, I used pre-trained model, but it was not as good as a unit, like my own unit. So maybe if I find one, it will be very useful to use.
Thank you.
Good. We have a question from, I guess, Martin or Felipe.
Yes. So the colon is kind of famously a very sort of convolved and folded structure. And so my immediate thought was, it would be so cool if you could kind of unfold it, right? So you could kind of reassemble it along the main axis of the colon. I mean, I know it's not an easy thing to do, but I just thought it would be so cool to do. And it might also help you, help the person who need to annotate data to actually have kind of understanding 3D slices in a very convolved space is hard. Whereas if you kind of unfolded it, it might actually make it a little bit easier, and you might highlight some sort of.
Yeah, that's a very good point. I will discuss that with Jan, but I think it's a very good idea. Thank you, Felipe.
Yes, and I think that Martin is also able to help you on the unfolding, since you have great experience in this area. Good. Are there more questions? I have a question also in the, and that's also perhaps I'll start with a comment, because it's typically what we see, this thing that you also do, start by downsampling the very nice high quality data that has been acquired, and it's definitely this forum is for sharing ideas, so we and sharing tools, such that we ensure to utilize the high resolution. I realize that it's not in the analysis phase, but also in the exploratory phase, where you need to zoom into regions and see things. It's good to keep the original high resolution, and we like have an obligation to utilize that, I would say. And then I also think that pre-trained models could be. I would also say that this is interesting, and especially perhaps it's a question for also other people. Are there someone that has some pre-trained models already that could be shared? So there's one question from Yash, and then we will go on to the next presentation. Yasha?
Hi, thank you for your presentation. I have a question. As I understood, you used MedSem to generate grand truths, yes?
Yes.
So do you think, is it reliable since it has not been fine-tuned on your data?
Since what? Come again? I didn't get it.
The model has not been fine-tuned on your data, so it is a pre-trained model. So do you think it is reliable to generate some grand truths?
Yeah, because this is a it's a semi-supervised techniques. So I'm just putting I'm just clicking on the column to segment it. So it's my role to determine if it's good enough or not. And so I think it's reliable, but as an automated technique, it's not, obviously. But yeah, since I supervise it, that's fine, I think. What's your point on that?
Actually, I think in some pre-trained models, the segmentation is not accurate, since they have some pre-trained weights, and actually you cannot accurately use them for your own data.
Okay.
So I think maybe we can actually fine-tune the models with some data that you have labeled them. So I think after this step, they will become more actually accurate on your data, and then your grand truths will be more accurate.
Yeah, for sure. But actually, the goal of segmenting it in this way is just to learn how to handle the real images in a model, to train a model. So just to see how it behave and how we can tune it just before we got more, ground truth.
The Institute for X-ray Physics, and the group of Tim Zaldit. And today I show you some results of our BM18 beam time of last summer, where we had a look at human temporal bones and the human cochlea. So just, I cannot change this. Yeah. So just to give you all a quick orientation, that what we see is the auricle and the outer ear. Then we have the middle ear with the 3 ossicles and the inner ear with the cochlea. And this is a highly delicate and very 3-dimensional object. So that is fully accessible if you don't have the opportunities to look at that. And here, so it's encased in a temporal bone, which is the hardest body in the human, the hardest bone in the human body. And so I show you a little bit later what that also means for imaging. And we had a look at different samples. So we had a look at the fixated humeral temporal bone as it is. We had a look at an optical cochlear implant in that to study optimal implant positioning within the bone, and also trimmed down the temporal bone to have a look at the cochlea, of which one we stained with the osmium tetroxide stain, and the other one we decalcified and put in ethanol. So it's a very weakly absorbing sample then, but has of course a good promise for phase contrast imaging there. So yeah, as an orientation, you all have seen the BM18 beamline. We also did some measurements at a micro-CT in-house. And as you already can see here, we used the HiP-CT protocol. So we mounted the samples in one jar and took the flat field either in a sample-free region in the jar or in a separate sample container with the same solvent. And so just to give you an idea of the sizes, we are somewhere between 3 centimeter and 7 centimeter in diameter of the sample containers. So, okay, for the brief for the imaging, it's a phase contrast CT. So every projection is corrected with the CTF scheme and then looks a bit more blurry, but it's necessary for the correct tomographic reconstruction. So just to show you some slices here in the figure B, you can already imagine the turns of the cochlea, and we did an overview scan of the whole temporal bone with every cavity and stuff around that, and also a zoom-in scan at the position of the cochlea. And yeah, since the whole temporal bone, we also have part of the middle ear. So this is the stapes, like the third of the 3 ossicles. I segmented this with the region growing algorithm. So somewhat semi-manual and to really see the cochlea and the 3 turns of the balance organ within the whole data set. And we also and the stapes, of course, and we also did like very quick rendering with Siemens Cinematic Anatomy. So for the temporal bone with the implant, we did not zoom in there, so it's just the overview scan, but we scanned that afterwards with the micro-CT in the lab. And as you see, with the high energies, we have little problems with beam hardening or metal streak artifacts. So it's very reduced to a small region, let's say that way, which you would usually have. But we also don't see then the fine soft tissue in the cochlea, which I will show you for sample at a later stage. But like for the implant positioning, that is reasonable. I prepared a video. I don't know how that works with frame rates over Zoom, but here you see the an unsegmented fly-through of the sample. So we go into the ear canal. Here you see a cochleostomy, so where a hole has been drilled in the cochlea where the implant was fitted in. And here now on the right side, we have a look into the middle ear with the 3 ossicles, the incus, the malleus, and the stapes. And the implant here goes into the cochlea. And if we cut it into the back, you really see where the stapes can like push in and out of the cochlea. I also did a segmentation to have a better look of the optimal implant positioning there. And here the implant was put into the basal turn of the cochlea. And here you see the single LEDs. And this is, of course, interesting. So the cochlea builds a tonotopic map. That means that different frequencies are resolved at different points in the cochlea turns. And now, if you know at what position of the cochlea an LED is, you can then map which frequencies are resolved at that point. And so for hearing research and for hearing restoration, this is an interesting model. And so also here, segmentation and was done with return growing algorithms in the cochlea. For a next sample, this is an excised single cochlea that we stained with the osmium tetroxide stain. And here you really see that it enhances the contrast for the nervous tissue, which is now shown here in a darker color, in almost black. And if you follow the shape of the cochlea, very interesting is always the basilar membrane with the organ of Corti, where the sound gets detected by movement of the membranes and the hair cells. And these are innervated with the spiral ganglion neurons, which are lay somewhere here. Yeah, with this nerve trunk of the auditory nerve that connects the tissue. So here there was also a nice rendering technique with a ray tracing framework from NVIDIA, which they formerly used like mostly for games. And here you can really see, so just to give you an orientation on the color map, which I didn't put here, blue is here again, a little absorption, and it goes over green to yellow to red, which is then more heavily stained part of the sample. And you can really see here on the right the part of the innervations of the organ of Corti. So to next, maybe more an outlook. So this is not as artifact free, let's say that way. This is a decalcified sample where we really had some problems with the finding a good energy. So we are like at high energy, so it's very suitable for large samples or for somewhat absorbing samples. And here we went to a part of the side beam to have a look at the sample and try not to oversaturate the detector, but it was rather challenging. But that is kind of our goal, to really have a look at the single hair cells in the organ of Corti. Maybe just as a quick note, on the left-hand side, it's a BM18 data set, and on the right-hand side, it's something it's recorded at the P10 beamline in Hamburg. So to briefly wrap it up, we have different contributions of the absorption and phase contrast in the different samples. So the large samples worked quite well here, although we had a little contrast for soft tissue there. And but for a bone and implant, it's a very fast segmentation. You could also do it, of course, with thresholding. But here, the cavities and the bone all more or less look the same. So really tracking the ones which belong to the cochlea here was a better option for me with the region growing framework. And so for little absorbing samples, we had some problems here during the challenges, since to find a stable configuration, and also at some point a chopper might be installed, and that would of course optimize that imaging regime there. So thank you for listening to the small presentation on imaging the human temporal bone. And for other projects, I also used some other segmentation and rendering techniques, but for these data sets here, that was really what worked for me here. And I'm open to your questions there.
Thank you very much for a very nice presentation. So let's give Jannis a hand. And now we have time for questions. So who has a question? I think there were some questions in the chat about what software you used. So, but let's go to the question. I think that Martin has a question.
Yes. So perhaps I missed it, but what is sort of the dream scenario that you hope to get out of all of these cool data sets? What is the kind of quantitative goal? Is there, what kind of measure would you ideally want to get out?
So I started my PhD really looking at small details in the rodent cochlea, and goal was, or one of the goals of our studies is to get an impression of age-related hearing loss by structural changes in the cochlea. So there we aimed at counting the hair cells and counting the spiral ganglion neurons, and that, of course, like if you want a high resolution, it works better on small samples. And so we had a look there at mice cochlea, and really there could also segment and count the spiral ganglions, which are more like small droplets in the cochlea. Here for this beam time, it was really to extend the approach to large samples, and also have a look here at the organ of Corti and the spiral ganglion neurons, which are soft tissue. So we had challenge here with the high absorption of the bone to combine that. So we went then more in a way to have like a model of the human temporal bone and the cochlea, which can be used in auditory research. For example, for simulations of the auditory pathway, so where the position of the cochlea can get mapped with a frequency that is suitable for that hearing position, and for to check for the implant positioning here.
Okay, I think in the interest of time, I think the rest of the question for Jannis should be saved for the discussion afterwards. So let's thank
So maybe just very quick to the question in the chat. Yes, I used VG Studio Max for the region growing algorithms, and for the other two renderings, which were unsegmented, the Siemens Cinematic Anatomy and NVIDIA Index frameworks.
We'll have Jakob Reichmann, also from University of Göttingen, present placenta analysis. So, Jakob, please start your presentation.
Sure. Can you hear me? Well?
Yes, we hear you well.
Great. Yeah, thanks a lot for the invitation to hold the talk. Also great to see so many familiar faces here in the chat and in the Zoom meeting. Yeah, I want to talk about 3D multiscale characterization of the human placenta by full field HiP-CT, and yeah, which we image on different kind of field of views with different instrumentation. So here on the left, you can see one example already for cinematic anatomy representation of one of these placentas. But how we got to this, I'll explain in a sec. So first of all, yeah, why imaging the human placenta? So the goal was to create an atlas of healthy human placenta to expand the understanding of the structure and also specifically on the vascular organization. So on the vasculature tree, which basically goes through your placenta. And this is interesting because this would be crucial to study effects of conditions and diseases which you have and which are based on the tissue morphology, and possible role of this changes in tissue morphology on birth complications like preeclampsia, but also on changes in the umbilical cord. And yeah, the second goal, like after the acquisition is done, is actually to develop efficient segmentation and morphometric analysis workflows to actually gain quantitative information on the tissue parameters, to actually be able to deduce information on these conditions and diseases. And the workflow was, first of all, the extraction of the organ, which, yeah, of course, happens in the hospital, but then the sample preparation, the acquisition, and of course, the segmentation and morphometric analysis of the sample. You can see here on the right one example for a prepared actual sample in a jar. So the imaging, so before the imaging could have been done, we extracted the samples and fixated them at 4% PFA for 5 to 7 days, and then had an ascending alcohol series to actually reduce the amount of water and replace it with alcohol to improve the contrast in our imaging. And then the degassing and mounting. So basically the preparation was done according to the HiP-CT protocol, which was already published by Walsh et al. From UCL, and with just very slight modifications. And then we did the imaging part, and which we did mostly in the BM18, but also in-house. I will show you some examples on that. And then we tried on the segmentation and quantitative assessment, which I've done in Copenhagen in cooperation with the group of Anders. And here is some example for one of the acquired data sets at the BM18. So we can see here that clearly the inner structure in your placenta and the soft tissue is very well visible of the entire organ, which is about 12 centimeters in size. This is a healthy control. And here we scanned it with 20 micrometer effective pixel size and acquired in volume of about 1.5 terabyte per volume, so per one placenta before binning. Here on the right, you can see a high resolution scan we did of just an umbilical cord with an effective pixel size of about 3 micrometers. Here you can see some of the results we achieved by just rendering, so without any segmentation at this point employed. So here on the left, you can see the whole placenta, where you can virtually, of course, slice through your volume since you have this three-dimensional volume at hand. And you can actually, by just using thresholding techniques, which basically rendering is based on, already segment out a major part of the vascular tree. You can see here on the bottom left. You can also go ahead and look for specific regions of interest, so specific structures which are of interest. In this case, we looked at hematoma, which appear frequently in placental tissue. And we found this here in our scan. You can see it right here, and rendered it here on top. And also, we actually physically cut it out of the sample after the experiment and did histological analysis to actually being able to verify that this is the structure we're looking for, and being able to actually segment this kind of lamellar structure in your hematoma. So here, this is the result which we can basically achieve with the entire placenta just by rendering and just by basically setting threshold parameters to segment our vasculature tree. But of course, to get quantitative measures, we need to segment this vasculature tree. And at this point, with the help with Patrick Møller-Jensen, also from DTU, we set up a UNet, which was pre-trained with already with data from micro-CT placental data, which was made accessible a few years back. And then we kind of extracted a 400 cube pixel subvolume and sparsely annotated the data on it using web Knossos. And with this data, we then we use this as a ground truth to train it on the entire placenta. So yeah, of course, we split the data sets and tested on different subvolumes. And then we also did, during this, when we trained our model, also random crops and data augmentation to actually make sure that the UNet learns as much as possible, and used some specific training laws and optimizers. And we saw that after some, let's say 180 epochs, we saw a significant drop in the validation, but also in the training loss after we actually trained it on the entire placenta, which the network didn't see before. And we can see as a result, so here on the bottom left, that the mirror segmentation of the large structures worked quite well. Here on the top, you can see a segmentation by Yashvardhan, Dahl, who used another MATLAB-based software to just segment the chords in the umbilical cord. And we can see here that from this unit segmentation, I'm not sure how the frame rate is working with the video, but that the major structures are here clearly segmented, but we also have a lot of false positives. And this is really the large issue we have here, and which, yeah, was the bottleneck we had. So that when you actually segment the structures, so when you actually visualize the structures you segmented, you can see it here, the rendered version, but in the segmentation, so the rendered version, of course, is also implemented, and you can see some of these structures. You can see here this red arrows, which were segmented, which were not visible in your renderings, but at the same time, you have a lot of false positives, which are very hard to actually remove because they're sometimes, they're like small, but when you try to remove connected components, you may also remove structures which you're actually interested in just because they're disconnected in your tissue. And this comes from the fact that the blood in your placenta is actually clotted, so that this blood gives you very strong contrast. But at the points where it gets thinner and thinner, the clotted blood actually stops to exist. So you have basically dark regions instead of very strongly, strong regions like you can see here. So this white, very strong contrast you can see is from the blood. But at the points where it disappears, it kind of, it's not feasible anymore for regular segmentation using just thresholding or seed-based segmentation. And this is also turning out to be an issue for segmentation using UNets. And you can see here an overlay that you have, some structures which we gained, but also some false positives. And besides of that, we also did, as I announced in the beginning, laboratory scans. So here on the left, you can see a whole organ which we scanned in-house. I think Jannis just showed you the setup we have downstairs. And here you can also see that this clotted blood is very well visible and gives you very strong contrast. But of course, at thinner kind of where the vasculature thins, it gets harder and harder to actually track them. And here on the right, we actually extracted the biopsy from the sample and scanned it at our high-resolution setup at P10 Genix in Hamburg at DAISY. And from this, we could also see some very interesting morphology and placental structure, which would be very interesting also in cooperation with the doctors when we talked about it, to actually segment it and quantify it, to actually gain some more understanding about this like placental architecture we have in the tissue. So the take-home message, we scanned our entire human placenta to create a human placental atlas, and had also several high-resolution data sets of particular regions of interest, which we extracted from our sample, and we could find some tissue abnormalities and do also histological assessment of this abnormalities. And by this, we kind of try to explore it with 3D image segmentation, as I tried you to show, and saw that there are still some challenges, some challenges to take. But this would be something for the outlook. And for things we are planning right now is firstly maybe try to adapt the protocol a little bit to shorten the preparation time of the entire preparation, but also try vessel staining, which we're trying right now with our cooperation partners to basically achieve higher contrast in our vessels by, for example, using iodine solutions or other vessel stains, which proved to be very effective in X-ray microscopy before. Of course, segmentation of the modeling, which I just mentioned, but also the translation to the lab to actually have a higher throughput in which we could do an analysis of more placentas to have a larger number N, which would be very beneficial to actually do like clinical studies and study abnormalities like you would do in a clinical setting. And by this, I want to acknowledge people at the RP. So Tim Saldit, Anne, Eva, and Susanne, who helped me with the project, but also, of course, our collaboration partners, specifically at the ESRF, with whom we did the experiments, but also at the DTU, who helped substantially with the segmentation. And of course, the IRP Beamtime team, you can see here. Thanks a lot for listening.
Thank you very much for a very nice presentation. So it's time for some questions. So who has the first question for Jakob? Felipe?
Yes. Thank you for the presentation, Jakob. It's very interesting. I have one question. Like you showed your training plot of your loss, and then there was a sudden drop. Could you go again on why that happened? I couldn't follow. Yes.
Here, this drop.
Yes.
So
exactly.
basically, this drop would be the. I mean, of course, Patrick could explain in more detail, but of course, I mean, this drop would be the point at which kind of the network started to actually understand the structures and being able to segment, or the training loss significantly drops, which is kind of common with this sparse annotations. So it means that it starts to understand basically what we are looking for. So this drop is actually something we're looking for, and basically means that the validation, like the loss function drops significantly, which means that the quality of our segmentation improves. Yeah.
Okay, thank you.
Yes, Claire?
Hi, thanks, Jakob. Lovely presentation. I was just wondering, again, on this loss and the loss, do you know, like, what's the accuracy? Two questions. So what's the accuracy the model is getting in the end, or the dice score that it's achieving? And kind of a related but different question. What dice score or other metric, what do you think you actually need to achieve to be able to answer the biological questions that you want to about the placenta?
Yes, for the first question, we didn't really yet calculate dice loss, so I couldn't tell you much on that in that regard, because we didn't have basically for dice loss. I mean, I would need to kind of manually annotate as much of the data as possible to actually be able to compare them.
to actually calculate the loss, which wasn't done so far, also because it's like a very large volume and manual annotation would be very tedious. On the second question, what we're looking for would be specifically, so this was a healthy placenta, and we also have some placentas which we already prepared, which have some anomalies, for example, this dysplasia, where we see a significant difference, or where we would expect a difference in the course of this vasculature. And by quantifying this, this would be very interesting to actually deduce on this kind of changes which would have, because for now it's only speculation. And so as much of the vasculature tree as possible, but by segmenting as much of the vasculature tree as possible, we hope to answer this question in the follow-up work. And also depending on because there are all other things you can use just by basically applying on your placental tissue. For example, calculating shape measures or difference, which I didn't present here, but which would not really require any segmentation, but just working on the gray values. And this would be also very interesting to see abnormalities in the tissue and differences between, for example, thick, like healthy and control. Yeah.
Hello, everyone. I'm Yashvardhan Jain. I'm a research software engineer for machine learning at Indiana University. And today I'll be giving a talk on hacking the human vasculature in 3D, which is a Kaggle competition that our team under Dr. Kati Bona organized in collaboration with Dr. Peter Lee's team at University College London. Our team works on creating the Human Reference Atlas, or the HRA, which is a comprehensive, high-resolution, three-dimensional atlas of all the cells in the healthy human body. The HRA provides standard terminologies and data structures for describing specimens, biological structures, and spatial positions linked to existing ontologies. It also defines how new datasets can be mapped to the HRA. It's authoritative, computable, published as linked open data. It is open for anyone to use, and it continuously evolves as new technologies become available. To learn more about this, you can go to the humanatlas.io website. And this effort is not isolated. It involves work from 17 different consortia, 250 plus experts, 1,000 plus publications. And one of those 17 consortia is the Common Fund's Cellular Senescence Network, or the CENNET program, which was established to comprehensively identify and characterize the differences in senescent cells across the body, across various states of human health, and across the lifespan. Senescent cells are cells that are unique because they stop multiplying, but they don't die off. The goal of the project is to provide publicly accessible atlases of senescent cells, the differences among them, the molecules they secrete, using data collected from unique multiple human and model organism tissues. Secondly, to create tools and technologies and a reference atlas of senescent cells in healthy human tissues. And finally, to unite cellular senescence researchers by developing common terms and classifications for senescent cells. One of the key challenges, which is something several teams in CENNET are working on, is the study of exosomes, membrane-bound extracellular vesicles used to transport diverse cargo to neighboring cells or even other organs. This cargo could hurt or help other areas in the body, and there's much active research on medical interventions using exosomes. Exosomes might help explain comorbidities in humans or how healing one organ improves the function of many other organs. Understanding the generation, transportation, and ingestion of exosomes requires data on the human blood vasculature, which is the main exosome transport system. And this is where the concept of the vasculature common coordinate framework can help. The VCCF database comprises 900 vessels, and it aims to localize each cell in the human body in relation to the vasculature that supplies oxygen to that cell. Using the experimental data, we can determine the pathway, throughput, vessel size, and 3D orientation in space of the human blood vasculature. And mapping the complex highway of blood flow will help examine exosome diffusion via the bloodstream to understand the role exosomes play in organ-to-organ communication, or where exosomes might aggregate due to branches or tortuosity of the vessels. At the single cell level, we can think of the VCCF as distances between different cells and the closest endothelial cell, or blood endothelial cell, which is what represents the blood vasculature. Here you have an image of skin data, of 3D skin data, where each cell is connected to the closest endothelial cell. And here you have similar examples from intestine and spleen, and this can be done using spatial proteomics or spatial transcriptomics datasets, running cell segmentation algorithms and cell annotation algorithms on them, and then computing these distance distributions. But how do we go from that single cell micro scale to the meso and the macro scale? And that's where the blood vasculature helps, because the blood vasculature permeates through all the different scales in the human body. This is also where the data from the Human Organ Atlas becomes extremely important. As many in the audience would know, the Human Organ Atlas Hub is creating an open access database using the synchrotron imaging technique called the hierarchical phase contrast tomography, or HiP-CT. It can scan intact whole organs at 20 micron resolution, locally zooming down to a micron or near cellular level without any physical sectioning. This volume data is being used for applications from anatomy training to helping understand our organs in health, disease, and as we age. But we need to go beyond just the images. We need quantitative data to be extracted from these images, and for that we need better algorithms. So why host this challenge? Why is the vessel segmentation a complex problem? So one of the big challenges with vessel segmentation is that the vessels in the images are small compared to the overall image, which leads to this imbalanced class problem in machine learning. We also have low tolerance for connectivity error, because more this error becomes, more noisy our signal becomes in terms of extracting quantitative data. Collapsing or blood infilling of vessels leads to issues with segmentation. There's large variability in samples due to natural human anatomical variation and post-mortem variation. And because of the novelty of the technique, there's very small training data that's available. And you can read more about it in our preprint that we submitted before the launch of the competition. So why not open up this problem to the machine learning community worldwide, right? So that's why we hosted this Kaggle competition. For those unaware, Kaggle is a data modeling and data analysis competition platform. It's the world's largest machine learning community, where businesses and researchers can publish data sets, and data scientists and machine learning practitioners can compete on the platform to produce the best models. It has over 18 million members. It hosts over 400,000 public notebooks. It has hosted over 500 featured competition and thousands of community challenges, and it has over 200,000 open data sets. It has been used by the NFL, Google, Department of Homeland Security, Lyft, and many other companies and agencies in the past to solve their challenges. As you can see on the top left, Kaggle has hosted thousands of challenges in the past 10 or so years that it has existed. And on the right, you can see that the community is truly global, where you have teams participating from all over the world. And Kaggle sees millions of submissions every year across its different competitions. So how did we set up the competition itself? We had 5 whole kidneys imaged at roughly 50 microns per voxel. We divided that set into 3 training kidneys and 2 test kidneys, one for the public test set and one for the private test set. We also provided some additional data at nearly 5 microns per voxel. All of this data was manually segmented by Dr. Lee's team at UCL. It was triple validated by expert annotators and required over 600 hours of expert segmentation time, which I hope helps highlight the challenge and the importance of solving this problem. We also provided some sparse labels for some of the data sets, and we also provided a baseline model using the NNUnet. The 5 winning teams in the competition were from many different countries like Japan, China, Israel, and Ukraine. And as you can see on the right, each team had hundreds of entries, where each entry is a single iteration of an experiment and a submission. So you can see just across the top 5 teams, we're looking at 700 different experiments that these teams did to be able to reach these results. The prize money was a total of $80,000 divided among the top 5 teams, and it was sponsored by Kaggle, CFAR, which is the Canadian Institute for Advanced Research, and Thermo Fisher Scientific. Now, during the 3-month period of the competition, we saw a lot of interesting discussion happen on the discussion boards, where different teams provided their solutions. They presented different data sets, and that really showcases this collaborative nature of Kaggle, where different teams with different expertise help each other implement different solutions. One such case was the marching cube loss that one of the winning teams used, which was recommended by one team, implemented by a second team, and then optimized by the winning team. But across the 5 winning teams, there are certain techniques, certain strategies that really help these teams create models that were able to get closer to solve this challenge. So one of the important things that all the different teams used were using intensity-based and rotation augmentations in their training data sets, but also in some cases at inference time. Focusing on boundary-focused loss functions like marching cube loss really helped, and post-processing the predictions to remove the disconnected chunks that are farther away from the vascular tree also helped remove the false positives. Coming up with different pseudo-labeling strategies helped teams use data sets that didn't have ground truths associated with them. And generally, the ensembles of 2D and 2.5D models perform better than using just 3D models. Kaggle community is a global community, and this competition was a success, where we saw 1,400 participants across nearly 1,200 teams. We saw 6,000 plus individuals that joined, 200 of which were first-time competitors, 5 of which ended up in the top 100. Across the 3-month period, we saw 32,000 code notebook submissions, which is 32,000 plus experiments that people had performed, which led to 500 public code notebooks, 700 plus discussion comments, and 200 discussion forum topics, which all adds to the overall knowledge in terms of solving this problem. It was truly a global challenge. We saw participants from 78 different countries, and that really helps highlight the nature of this community, the idea of researchers opening up challenging problems to the community and getting state-of-the-art models in a collaborative way. And that showcases this power of citizen science that we can use in future to unlock more better algorithms for like different challenges. Finally, I'd like to thank everybody that was involved with setting up this competition, from our team, from the team at UCL, from the team at Kaggle, and to all the 1,400 Kagglers who actually participated and the larger Kaggle community. If you'd like to learn more about this competition, you can go to kaggle.com/competition/blood vessel segmentation, where you can access the data sets, learn more about the competition, and you can access the discussion forums and everything. And finally, I'd like to thank the organizers for giving me the opportunity to present our work in this workshop. And thank you everyone for listening. If you have any questions or if you would like to reach out, please feel free to contact me at yashvardhan@iu.edu. And with that, I conclude my talk. Thank you.
Very nice. So thank you for that presentation. And I think that if there's a question, it should be addressed to Claire. But I can easily start, because I know that you mentioned that the data is, of course, the hierarchical phase contrast data, but that was not what was the main focus of the participants. So how did it go? Did they utilize the hierarchical nature of the data?
So actually, they didn't utilize it in the sense of using it to predict labels from high to low resolution or anything. A few of the teams did just take the high resolution and put it into the whole training data bag and just used it as another data set. No one really leveraged it in the hierarchical sense. But then I suppose that's also was challenging because we didn't provide the registration coordinates for it in the metadata, because yeah, it would get too complicated.
Okay, so there's still some unused potential there, and I guess that the training data is available on the Kaggle site for everybody to use.
Yep, available to freely download, and the winning models are all there as well. So kind of building on the open model idea.
Great. Are there other questions for Claire? Otherwise, thank you for setting up the contact and for getting Yash to send his presentation. We hope to see him in real live next time. And we will continue with the presentation of Felipe. So Felipe is locally here from DTU, sitting in another room right next to. So Felipe, please start your presentation.
Yes. Okay. Thank you, Jannis. So as you're saying, my name is Felipe, and I'm research software developer here at the King Group. So last year I joined the group, and then our idea is to bring all the tools and software that it's being developed in this more academic setup closer to the users. So then making everything that we can develop more usable and easier to be reached. And instead of having slides, I thought it was a good idea to do live demos. As we are developing software, like it could be nice to run the software that we're doing live. And so that's what we'll do. So I'll share my screen, and we'll see how we go through a few examples of the things that we've been developing here towards the last year. I'll just share my full screen, and I think you see yourselves now. So just as like an introduction to it, so we're a part of the King Center, right? And one of the things that we're developing is a web platform that you can easily access going through the King web page, and then you go here to access the King platform. So this is a web platform that the global idea is that it can allow users to more easily interact with their data, focus on 3D volumetric data. And the idea is that eventually this will be easily accessible. So users will be able to simply register and have access to computational resources here from DTU. But the part that I want to focus a little bit more that can actually be used right now by all of you is the Python library that is behind the platform that we're developing now. We're calling it King3D. So the documentation for the library can be found here. And I guess that if you just go to Google and then you just type there King3D, it should be, if not the first, one of the first results that you find is the documentation for the library. So what's that? So this is a pip installable library that you can very easily just do a pip install King3D. And it should the idea is that here we are building up tools that help people more easily do 3D image analysis. Sometimes it's not so easy to have algorithms that are focusing on volumetric data, and we have lots of challenges that they come together with volumetric data, right? So we want to have something that we're sure works well with volumetric data, and for example, like can handle large volumes, or we're sure that all the operations are really be done on 3D and not like on 2D slices and things like that. So I'll run some notebooks here. First, I'll just go through a first notebook with some easy examples, just to give you the feeling of what we are developing for the library. So it should be very easy to install. As I said, you do a pip install King3D, and if you run import King3D, you should have the library ready to use. We have some examples that come together with the library, some examples of small volumes that help us start exploring things. So here I just imported one of the examples that we have. And one of the focus of the library is to give you tools to visualize and easily visualize your data. So for example, by running King3D slices, you can ask for a number of slices from your volume, and then it creates a nice grid that you can have a quick overview. Or you can call the slicer function in your Jupyter notebook, and then you have a very easy interactive slider that you can go through your volume. And then this can be really helpful to explore your data while you're doing some processes. Also, it's possible to do volumetric visualization right there in line in your Jupyter notebook. So here is the same data set, right? And then it could be a little tricky to understand the data just by going through the slices, right? But then having a volumetric visualization that you can interact with, we know that sometimes this really helps us to get a better feeling. And here we are using K3D to do the visualization, and this contains all the things that you usually expect from a volumetric visualization. And the idea is also to give the basic tools that you can start doing all the process that you need to do with 3D images. So for example, here we have like a 3D top hat simple operator that can remove background objects from your image. So it's quite handy to be able to do that in one line and being sure that operations are being done on 3D. Or also we have like filters that we're working on making them optimized for 3D volumes. So for example, here I just did a small Gaussian blur on the same data set. And then of course, like we can very easily make a threshold there and like try to segment this data set here. And very easily we can do a 3D watershed and visualize the results of it, because we could see that from here, for example, you have some objects that are connected, and then, for example, in this case here, and then the simple 3D watershed, it's what we would need to get our individual components there. So in this case, we found 559 objects in this data set. And then the idea is also like to bring some more complex algorithms. So for example, here I'll just show a structured tensor analysis. So I'm loading another data set, and I'm using here another function that we call orthogonal that give you interactive sliders for each of the axes of your data. And the idea here is that we could do some structured tensor analysis to check the orientation of these structures. And very easily, like in one line, we can call the structure tensor, do the calculations, and bring a visualization tool that allows us to actually see and interact with the data and very quickly get insights from what we're doing here. And I want to show something using human organ data sets. So I just, to let you know, I was exploring using the Human Organ Atlas page, and I found this data set from the heart here. And then I wanted to have a quick overview of what's happening there. And then I wanted to use the King3D library. So I just downloaded the zip here and extracted in a folder. And then I can, in this other notebook, so this is the path where here locally in this laptop, I extracted the folder. And then I can use our data loader to load the data. And because I'm calling a virtual stack, it's using Dask to create a virtual object using chunks of this data set. So the data set is not directly loaded to disk, but when it's using Dask, it allows me to just use the data when needed. So on this other cell here, I'm just then getting the data from one specific part of the volume. And I hope that you can still hear me because this is parallel and uses all the cores that I have available. So I'm not sure if you can still see the Zoom meeting while I do the things you can. Okay, just checking. Because the idea is that we wanted to do this as parallel and as fast as possible. So because it's a smaller cropped region, it should finish loading soon. And then we can use the visualization functions that we saw before on this cropped region of the Human Organ Atlas datasets. And then again, like this, I don't it's a normal laptop. We're not running in a cluster, so I don't have huge amounts of memory available. But because we can use this Dask object to create this virtual stack that we can just use as a normal Python object, and then we can just load to memory the parts of that bigger object that we need to process the data. Mm-hmm. Maybe I should use a smaller crop of this object.
While we're waiting for that to load, I was going to say the crop,
yes.
The crop bounding box that you've chosen. Did you choose that on looking in the Human Organ Atlas at the NeuroGlancer? Did you pick that crop out because of a particular feature that you wanted to see, or was it just a random crop?
This I was exploring by myself instead of using the NeuroGlancer. But I see that it's really nice that on the website you have directly linked to the NeuroGlancer, and then you could use it to explore the data there, which is very, very nice. But here I was basically testing and by myself trying to find some structures there. So I think it's finishing. Let's go back to the notebook there. So I see that I have a cropped region. So then I can call the slicer in this notebook, and then here we have the region that now we loaded to memory. And even though it's we're used to scroll through images like that, it can be a little bit hard to understand, right? What the structure actually is. So we can call the volumetric visualization that we saw before for this specific part. So this will take, I think, a few seconds, and then it's the last thing that we can see together. Yeah, and then we're using this library as for the moment with building blocks of functionalities to process 3D images, but then the idea is to start more and more being efficient on handling large data sets, because this is one of the biggest challenges that we have, right? How to handle big data sets in a way that's efficient enough. So then here we have the volumetric visualization that we just process, and then we can interact. And then now it's a little bit easier to try to figure out what's happening there. So it looks like it's a vessel, and then there's some structures there that I'm not in position to tell what they are, but looks like there's something happening there. And sometimes those initial exploratory aspects are really good because they allow us to quickly interact with the data. And the idea of the library is to bring this closer to the developer, so that the developer, like for example, while doing their Jupyter notebooks or any other Python code, can very easily interact and explore the 3D datasets. So I guess because of the time, that's what I want to show you. And then if there are any questions, we'll be happy to talk about it.
Thank you very much, Felipe, for an interesting presentation. Of course, I think it's interesting because I am also owner of that project. So, but thank you very much. So are there more questions for Felipe? Yes, Joseph?
Yes, thank you for this very nice presentation. I had one question is, can you, when you want to do 3D processing on like a huge data set, do you have to load your data and do the process, or can you do it on chunks?
This is something that we are exploring how to be the most efficient possible, because you can process chunks, but at some point you might need to save everything to memory when you need to compute that. So we are working on how to use the disk more, even though of course it will be slower, but sometimes like we just want to be able to process all the data, regardless of its, let's say, takes more time, right? But this is something that we started to explore now using Dask. So we'll see how to use Dask and be able to, let's say, just have your operation that you know works on the crop, and then just do that on the full volume without worrying. So that's the final goal, to simplify the whole operation.
Yeah, exactly. It's the same like for the orientation. I use your orientation algorithm. It's the same like if you want to run it on the whole thing, at some point, yeah, you need to do it in chunks, because if not, it's not possible.
Yes, yeah, that's true.
To the final presenter, which is Andrew Cook, and he will be able to tell you something about what we see in the heart images. So please, Andrew, start your presentation now.
Thank you very much. I'm just going to share my presentation. Can you see that?
Yes.
You see the screen? Yeah, great.
Yeah, there's something. So if you minimize that one, that's
just get rid of that. Yeah, it's annoying when it's you get a black box.
That's fine.
Good. Okay. So there's some very nice presentations and ways of analyzing the HiP-CT data. What I'm going to present really is the challenges in segmenting parts of the heart, in particular the cardiac conduction system. So really, I'm as much as anything, I'm looking for ideas, extra ideas, and the way we may approach this, other than manual segmentation. And you'll see at the bottom, there's actually quite a number of people that are sort of working on this, including Marion and Joseph and PhD students, Speisch, etc., etc. So we really, I think I'm really after ideas. The heart, of course, is a very hierarchical organ, as are many organs. So the myocardium going from the whole heart down to sort of myocyte level, that's certainly interesting hierarchy. I think we're discovering more with the HiP-CT data on that. The vasculature, the valves, but I'm going to focus, of course, on the cardiac conduction system, and that really starts with the innervation of the heart, with the nerves coming into the atria and around the arterial roots, then the nodes, the SA node and the AV node, going all the way into the Purkinje fibers, or the so-called Purkinje network. So it's really quite a hierarchical system, obviously crucial for early survival as an embryo. You need a functioning conduction system. The heart needs to function correctly, and then, of course, throughout life. And I'm going to focus primarily on two areas: the sinus node, which is within the right atrium. So if you like, the atrial conduction system, and then the ventricular conduction system. Now, actually, that starts in the atrium as well, starts in the right atrium as the AV node, runs through what's commonly called the HIS bundle, but we now call the non-branching bundle, into the bundle branches. And the problem is that we sort of have these 2D representations of what is a very three-dimensional structure. So this diagram, which is one of ours, is actually a bit of a misrepresentation of the real structure. So that's really the challenge, or one of the challenges, at least, in terms of the conduction system, understanding the three-dimensional structure. But let me show you some of the others. So in terms of the sinus node, first of all, the only way that we can identify it, and the way surgeons can identify it during operations, of course, is by trying to identify the artery, this tiny little artery, so sort of 1 millimeter artery that runs up to the region of the SVC, so the superior caval vein there. Tiny little artery runs up over the roof of the right atrium, and that will supply the sinus node. So if we cut across that, so we cut across that region, you will see, of course, surgeons don't do this, they wouldn't do this, but if we cut across that, you'll see there's this tiny little artery there, which is supplying the sinus node. And just lateral to that, so in this region there, if I pause the video, is where we'd find the sinus node. Now, clearly, you can't see it in this specimen, and you wouldn't be able to see it really very easily with surgery unless you can see the artery. So HiP-CT data can certainly help us visualize that and also sort of understand the variations in the sinus node. This is one of my videos, and it's not particularly good, but I think it illustrates the point. So this is a control heart, and we're going to zoom in on this upper region here, which is the area where that little artery is. And it'll go to high res in a minute. So this is the artery to the sinus node. I'm just going to let that play for you, because you'll see that the only way of recognizing the sinus node is this condensed cluster of cells. So this dense cluster of cells with fibrous elements within it, which surrounds the nodal artery. So you'll see that actually it's quite a lengthy structure in this case. So here it is at the top. And that's very different to the working myocardium, what we call the working myocardium underneath. So this is the atrial wall. So certainly you can recognize this if you know the histologic features of the sinus node. And this is what it looked like histologically. So we know that it's got this very dense appearance with the blue elements of fibrous elements and quite small cells. And we can then recognize that with HiP-CT data, and we can see changes in, for example, fatty infiltration. Here we can see changes to that structure. But I think you'll appreciate that it's quite a challenge, perhaps, to recognize this, this area, other than by manual segmentation. At least it is for us at the minute. And the additional challenge, of course, is that when you get abnormalities like fatty infiltration, you kind of lose a little bit of that density of the cells around the SA nodal artery. So, it's fine to recognizing normality, but then you've got to be able to apply the same techniques, of course, or similar techniques to abnormal situations. Now, this is just a very basic segmentation, of course, it's one I did on the HiP-CT data, but you'll see it's quite an odd structure. So it's actually quite difficult to image because you have to get lots of high resolution areas, and it's quite lengthy. It has a tail. and it has a broader head at the top, and different components of this are active at different times of the day. So the head when you have a faster heart rate, and the tail when you have a slower heart rate. So, you know, it's very important and sort of interesting to understand that. So that's the sinus node, probably the, you know, the one we focused on less, I suppose, in the HiP-CT data so far. If we move into the AV node, again, the only way of really recognizing this is to use anatomic landmarks currently. So we use the artery that supplies that node, artery to the AV node, which in most people arises from the right coronary artery, but in a small proportion arrives from the left. And of course, that's important in terms of if you have blockage of one of the other of those arteries. And this is what it looks like. So this is a left dominant pattern, and this is the circumflex coronary artery running all the way around the mitral valve. And then you'll see what we try to recognize, or what surgeons can recognize, which is this tiny little branch here running upwards into the heart. So this is the avinodal artery there. And that runs right up inside the heart. So it actually runs in a sort of groove of tissue up to this point, which is where we'd find the AV node. So at the top of that part of the tricuspid valve, just in there. So as you'll appreciate, you know, you can't see it. Conduction system is invisible. We just focus in on that region. Again, the conduction system is completely invisible to the surgeons. So all they use, or what they use, is the landmarks, which is this triangle here called Cox triangle. And the AV node would be positioned within the atrial muscle here, just in this region. So it is quite difficult to look at surgically. It's quite difficult anatomically as well. The traditional way of looking at this, just to put things in context, is you take blocks of that area, as you would for the SA node. You'd need about 6 blocks for the AV conduction system. You would cut that into about 2,000 or 3,000 sections. You'd then have to stain 1 in 10, because that's basically all you can deal with. And that would be in a specialist sort of research setting. So you end up with thousands of sections per heart that you then have to manually reconstruct and try and put back into the context of the whole heart. So clearly, HiP-CT data is much better for that. Let's go back one slide, actually, if I can. No, it's not going backwards, but anyway, this is the region of the conduction system in the HiP-CT data. So it's a small area. It's a very small area of the heart. You know, this is only a few millimeters across, but clearly it's crucial for correct conduction. And what you appreciate is that it has to be connected. So you've got these strands which are all connected together, but in different orientations. You've got some coming straight at you in the plane of the screen, others coming downwards into the left bundle branch. And then, of course, you can track it all the way back. This is the region where the left bundle branch is just originating. And I'll show you some more data in a minute, but we can go backwards in the heart. So these are sort of a step through different sections. And this would be the region of the AV node identified by its artery. You can see it's beginning, the cells are beginning to sort of condense here, but they are only subtly different from the surrounding atrial myocardium. They're a little bit more condensed, a little bit more compact. Very important split in the artery, which helps me identify this region. And then as we move sort of deeper into upwards, in fact, in the heart, you'll see that we get an area which where the AV node becomes a little bit more condensed. So we're coming up to that area here. And the reason we can begin to identify that is because we've got this fibrous tissue sort of forming a cap on the top there. So you'll appreciate the cells are slightly smaller, slightly more condensed. The wiring of the heart here, the conduction axis is now running through the fibrous tissue. And you see it's sort of fragmented here, which is of interest. It's also connecting to the ventricles, which also is of interest in terms of analysis and conduction abnormalities. And then it's coming down into the left bundle, which is here, coming off here, starting to come off. And then it will also run into the right bundle branch. So just let that play for a little bit, because I want you to appreciate the connectivity of all of those components. So here is a sort of narrow region of the left bundle branch, but you'll see it gets actually quite broad here. Oops. So at the end of that video, you could see the left bundle branch sort of segmenting, sort of getting broader. I'll just speed that up so I can show you that bit. But and also you'll be able to see the right bundle branch coming off. So just scroll through this again. So there's you'll see there are vessels running through the fibrous tissue, which is new information. I've not been able to see that before, so I'm just scrolling through as fast as I can here, but we will get to the bundle branches hopefully in a minute. And you'll see, so this is, I've created this video by looking, by sort of going through the sections. Here's the left bundle branch. So that is slightly thicker. And then hopefully we'll begin to see the right bundle coming off. And again, it's very subtle. So you'll see a strand coming down here, and then it begins to sort of condense and become more of a cord-like structure. So it's a bit sheet-like at this stage. And then you see it's coming down and it's beginning to sort of condense and become this, which is the right bundle branch. And that aligns with these attachments of the valve here. But it's very subtle. And I think, it's really sort of by tracking it section by section that you can follow these, rather than sort of being able to identify these as specific cell types. So this is sort of data that's in press at the minute. It's, you can identify these structures in the normal heart. It's sometimes easier to detect those in abnormal hearts because fatty tissue, when it surrounds these areas, can sort of highlight them, if you like, isolate them. So this is the AV node in an abnormal heart, for example. And the normal connections with the surrounding myocardium are sort of stretched and attenuated. And that, of course, can lead to rhythm problems. So, it's a challenge, but actually it can be sometimes easier in abnormal situations. And this is what it looks like on histology. It's not much easier. So of course you can do some basic segmentations. This is one I did of the control heart. This is just sort of AV node, bundle of His, and part of the left bundle branch. It's not really showing the right bundle branch, just showing the origin here. But all of these connections here with the underlying myocardium, so in this region, and in fact there's some at this side, are very important in terms of arrhythmia, so problems with cardiac rhythm. So that's really where we're at. We're the process really of manually segmenting, and Marion, Joseph are doing this, and Vaish is doing this in some other abnormal hearts. The even more challenging part, of course, would be to look at the distal system. It's called Purkinje system, but that sort of comes from animals, and there really isn't much information on that. The best diagrams were produced in the 1900s, so 1906. We think these were produced by injecting Indian ink into the sheaths surrounding the conduction system. So, of course, there's always a bit of error with that. But really, we don't have similar data, and the distal connections of the conduction system with how they ramify into the myocardium is of interest for understanding, of course. So HiP-CT data, we can actually see those. We're sort of exploring that at the minute. And if you take a section, for example, across this region in the control heart, it's this region. These are the cells that we're talking about. And again, it's sort of very subtle. So if I let that play, you'll see there is a layer of cells which are running in a different orientation to the underlying myocardium. They are slightly vacuolated, so they've got sort of holes within them. But it is a sort of sheet of cells that's running just below the endocardium and above, of course, the myocardium below. So again, I think tracking these cells, these have to be continuous with the rest of the conduction system, and tracking these cells, I think, is the real challenge here. So, I'm opening that up as a challenge as much as anything else. It really is a tricky thing. We've been thinking of ways of analyzing this, and open to suggestions really as much as anything else. Thank you.
Yes, thank you very much, Andrew. Very exciting to get a walk through the heart anatomy. And also highlighting the value created by the human organ atlas data. So it's actually you are a very good example of showing that we now can see things that no other technique can actually do. So I think this is really cool. So thank you very much for that.
Pleasure.
You shouldn't be stressed about us running late, because this is the last point before the discussion. So let's open for questions first for Andrew. Yes, Felipe.
So it's not exactly a question; it's more it's a comment, because I got me thinking about this during your talk, because it's really nice. It's super beautiful to see the structures and seeing that much detail. But then I was thinking that right now, my life is depending on those structures, that they look so fragile. And then I started to get a little worried. And then I think this is
Don't get worried.
This is one of the nice things about being able to visualize ourselves, right? So then we can see how we really work, and then how sensitive things are and how fragile we are. In fact.
Yeah, no, it's absolutely true. I mean, yeah, don't get too worried, but it is, I think part of the value of the HiP-CT data is in the visualization, isn't it? As you say, sort of us helping everybody understand where the wiring of the heart is effectively. But then also, as you saw in some of that data, understanding the regions around the conduction system. So we know that fat can begin to infiltrate from your 50s onwards around that area, but fat is inflammatory, and so that will affect the gene expression, likely affect the gene expression around the conduction system. So I think, yeah, you're right. Sort of understanding the gross level and understanding the three-dimensional structures, and then sort of putting it together with gene expression, etc., is really crucial. We need to sort of put it all together, don't we? And for surgeons, they're training using those drawings I was showing you, or showing lines drawn on pictures, of the heart. And actually, we need to be teaching them in 3D, with virtual reality, I think, to help them understand things a bit better. I don't know, I mean, I know Baish is on the call, but, we've only found, the only way we found of doing this is sort of manually segmenting, and there's a bit of thresholding, isn't there, Baish? So we're sort of using a combination of techniques, but it is sort of manual adjustment of sort of thresholding. But I'm sure there are other ways of doing this.
Yes, Joseph.
Yeah, just a comment that your video made me think, actually. It's true, for now we just do manual, but it's the main difference between the conduction system cells and the surrounding cells on the myocardium is their orientation. So we should really try to do soon to do an orientation analysis of this area to see if we could see something different than on the images. Maybe we can feed that into a model, or use that as to separate both the bundle and the surrounding cells?
We did, in fact, on the easier bit to recognize is the bundle of His, because it's completely surrounded by fibrous tissue. And we did a quick structure tensor on that region a while back. We didn't take it, haven't taken it much further. And you can see a difference. I mean, you can see structure tensor, but I guess, you know, then doing that on the whole system might be feasible and might help. But there are some regions, I mean, as you know, Joseph, you kind of need to be looking in really high res, don't you? Otherwise you can miss the connections, or you can miss you know, parts.
And also sometimes you have to pass through the slice to see like where it is, because the orientation is not the same. So you can, it's not easy.
Yes, and I guess that tools for structure tensor analysis to get the orientation, and tools to track the orientations, and also then to unfold and like interact in this way is something that's in great demand. So it's not necessarily a fully automated deep learning algorithm, but just to visualize the structures, you need to segment them out locally, as you show. So Claire?
I think in terms of a comment on trying to decide on like an image processing technique to use, and I think it's the same for a lot of the talks we saw today, is what's the final biological quantitative output you want out of it? And that has a really big dictation impact on what you use. Like, are you looking to extract the morphology of it? Do you want to know, the volume compared to volumes in others? Do you just want to know like the start and the end points? And I think that comes with a lot of stuff. So what process you use depends on what the question you want to answer is, and how much precision do you need in the answer of that question for it to be useful.
Yeah, I mean, of course, there's obvious sort of wins there in terms of, you know, directing redefining the landmarks, let's say, for surgeons, because I'm not entirely convinced that they're 100% accurate, which is worrying, of course. But I mean, you know, they're avoiding the rough area as long as they avoid the rough, you know, the rough region. So that's one thing. I mean, we can get better understanding of the connections. So what's normal, what's abnormal. So gross morphology, the sort of, you know, what some people consider abnormal connections are probably part of normality. So that's relevant to sort of pacing, you know, pacemakers and pacing individual bundle branches, for example. But the other thing I didn't show is you can use that in addition to the map of the myocardium, so the orientation of the myocytes, to simulate the conduction. So, you know, if you see the conduction in the AV node, for example, and you know the course of the bundle branches, the Purkinje fibers, and then the orientation of the myocytes, you can actually simulate the whole conduction, and then you can work out where would be best to pace. I mean, we're showing normal stuff here, but you know, in abnormal hearts, it can be tricky to know where it's optimal to pace. So yeah, all sorts of things, exciting things you could do. Potentially, yeah. But it all starts with knowing, I suppose, just mapping it out, doesn't it? And knowing the morphology, yeah.
Good. Are there some more questions for Andrew? If not, I think we should thank all the speakers once more. So virtually by where is it? Reactions, and then we will. Oh, that was the wrong one I had there. I couldn't find mine, but I'll do it then like this. I think it's been really exciting to see the progress that's been made and the different approaches to the analysis of the structures of the human organ atlas data. So it's clear to see that that there are different levels. So we've seen visualization needs to do the initial explorative studies. We've seen some segmentations based on simple non-learning approaches, and we've also seen some examples of unit segmentation, and it's clear that when you use this kind of voxel-wise classification of data, you'd see fragmentations. You'll see things being split up and does not necessarily follow exactly the anatomical structure. So there's clearly some unsolved problems here, and also some need for extra learning techniques. And then there's the need for exploratory tools. So being able to go in and visualize structures, unfold structures, and see things. So I think this shows that there's a lot of knowledge, and there's also a lot of inspiration to gain from these examples. So I think that at least I got a lot of ideas from seeing these things, thinking that there are tools available, and we should share them and use them for doing better analysis of these structures. And this is also what will lead to the next step in the series of these workshops. So I don't know if there's some comments or some questions or things from
I have one
that you've seen today. Yeah, so let's start with Martin.
I was thinking now, also during your presentation, Andrew, you said that traditionally the way to image the heart would be to have 6 different blocks, and then you would slice them, I suppose, in different orientations or what. And along these lines, the question is then if we can do virtual imaging. I mean, we can do virtual histology on a single heart, then you can map it out on a single control, right? Healthy heart. But would you need to have more? I mean, how much variation do you have between? So would you still like to have 6 hearts, or more or less? What would be ideal? And I guess the same question goes for the placenta, I mean, all of our other organs basically. How many do we need to have a good atlas?
Yeah, that's a very good question, isn't it? I mean, there are variations, of course. I mean, there are general rules, but the exact course of the bundle of His, for example, can be completely membranous, can be, a little bit deeper within the myocardium. So we sort of know that. And you may catch one of those in 6 samples, but in 20, you're probably going to get a bit more variation. I don't know if that's going to change sort of tracing or tracking the conduction system, because as long as you start, let's say, in the bundle of His, you could track backwards and sort of forwards from that point. And I think that's the key, is sort of tracking the streams rather than perhaps sort of trying to identify particular components individually. So yeah, it's an interesting question. So I mean, as many as possible, of course. But yeah, I think we need to be sort of 12, 20 for controls, really. 6 is minimum, I would say. Yeah. I don't know what others think on that, on other organs. But it's a good point.
I think definitely in the kidney, the vascular tree has huge variability in it. I think we're going to need a lot more before you can understand kind of the range of, and I guess it's that thing of what's the range of physiological, like normal, and what's pathological. And it's just trying to understand where those bounds are.
Yeah, and also, how do we translate that to a language that we can communicate and quantify? And so when people look at the same structures, that they to tell the difference and so on, I think the quantitative tools for that is not necessarily already existing. I don't know, because one thing is describing it, and another thing is actually measuring. Yeah. So.
I have another question, also then related to measuring more of the same type of sample. I don't know how much experience you have already now in the HOAHub on doing more of the same type of sample. But then the question is also from the data acquisition point of view and the data quality point of view, how similar are they? I mean, do you always get the same grayscale range, or just how dependent is that on the exact embedding of that particular sample, if it's a different person doing it, or when you come back to the same beamline a few months later and then you're doing the same experiment in principle, but things have changed a little bit, and then can you compare the images, or what is your experience in this respect from the BMA team and the HOAHub?
Claire? Rather than me. I didn't know if that was directed at me, sorry. But
take Paul or Joseph or somebody who's a BMA team.
Yeah.
Paul, we don't hear you.
You're muted, yeah.
No sound from you.
Maybe Joseph or Teresa or Hector or somebody wants to jump in.
What I can say is that generally for every individual sample, the contrast is adjusted individually, let's say, and the reconstruction algorithm does it on its own. However, we do know which parameters we usually use for reconstruction for different organs at different resolutions, and we try to keep it consistent. But of course, this can change a little bit depending on the issues you might find. In terms of post-processing effects on that, I don't know, Claire, for your kidneys, if you saw something related to this.
Yeah, we see a lot of variability in the windowing kind of that's needed across the organs, and that's a bit to do with the variation in the samples, the variation in sample preparation. Also, when we're collecting over a long period of time, BM18 is a beamline under development still, right? So it changes, and we get new optics, better detectors, and all of these things change. And that's one of the challenges with creating these kind of machine learning models in particular. They have to be able to generalize to these types of changes.
Right.
Yes. Other comments? I have a question to all of you. So if there's something that you've been inspired by the talks today, or have some ideas for the next workshop, do not hold back, and please come with some suggestions for what we could do. So if there's some comments in that direction or something, I would be happy to. There's one thing in the you're saying HOAHub Kaggle, Claire. So is that you're thinking about making several challenges in the same way as you've done?
Yeah, I just think it's an interesting thing to explore as a group. We have really interesting data across lots of organs, a lot of expertise. It'd be a really interesting, exciting thing to put together as a collaborative effort. I think David.
I think that outreach to the AI community, yes, definitely.
Another thing that might be interesting is that in the next few months, we really want to improve access to our data in chunked file formats. So at the moment, you get them in the JPEG 2000 slices, which are really slow to load, which you kind of saw in Philippe's demo there. So we have them in a chunked file format, which is called ZAR, which is the data compression isn't as good, but it's way better if you want to just load subvolumes of the data, and it's much faster to read into memory. So yeah, maybe at the next workshop I could do a quick demo on that. That might be of interest to people.
Definitely. So are there any state-of-the-art analysis direction? So like everybody's talking about deep learning, so I guess that's a good topic. But I think that like the data for deep learning is very important here. We don't like a lot of deep learning research is based on benchmark data where you have all the annotations together with it, and we have a lot of data here, but we have no annotations, and getting the annotations, I think, is a crucial step. So any views on topics in this direction? What analysis methods could be interesting? If you have some, you can either say it now, or we can do it offline via email later. I think that the crowd is getting quiet. That's not what happens if you collect this many people. We were, I think, over the presentations here, we were almost 50 people at some point. So it's really been a success. This first workshop in this this work area, and I think I will then round it up here and just wrap up. By saying that you will all be informed about the next workshop, and we'll find a date. We just need to coordinate to make sure that it doesn't overlap with other activities. But we aim at some time in November this year. But the announcement for it will be, so you get the date into the calendar, will be before the summer. And then, I don't know if there's something I forgot to say.
I would like to ask about the recording of the meeting.
Oh yeah.
Will you share it with us?
I will.
Can we share it openly, or will you put it?
I will at least ask the presenters. So if there's anyone that right now objects to sharing what I've recorded today, so that's the presentations and this discussion, they can say it now. But otherwise, I'll ask all the presenters just to make sure that they didn't show something they don't want to get out. But otherwise, I think we should share it openly.
We could also, if it's of use, that we have the YouTube channel for the HiP-CT technique. If people agree that it's openly shareable, it could go there as well.
That would be great.
Yeah, I think that's a great idea. Good. So I'll just round off with saying that I think it was really exciting to see the progress, and but there's definitely also still a lot of things to follow up on. So I think that before the next meeting, I at least see quite a lot of bilateral communications I'd like to have to discuss things and discuss also how to follow up on things and how we can collect and share the findings that we do, and especially the useful experiences for getting as fast from the data acquisition until we have some quantitative results of the data. So with that said, I would like to thank you all for listening in, and there was luckily no major disturbances during the presentations. So thank you for that. And I'm looking very much forward to seeing you all here in Copenhagen in half a year from now, in the nice November weather of Denmark. But that'll probably just keep us indoor. So looking forward to seeing you, and thank you for now.